So yeah. So this is on class is on this chapter is on human genetics. It says human molecular genetics. So what I thought that before going into genetics, I don't know if you have seen before or not, but what I want to share with you is the first paper that was published by Watson and Crick when they discovered the structure of DNA. So this chapter is on, this chapter is on human molecular genetics. So I thought to share with you the original paper that Watson and Crick wrote and is published in Nature in 1953, right? In 1950, on April 25, 1923, 1953, I apologize. This paper was published in the scientific journal Nature. And the paper said the molecular structure of nucleic acids. So if you look at here, this is just one page paper. So it's very short paper. But this paper created history, and this paper also created controversy. So briefly, I will just mention controversy, and then perhaps because Rosalind Franklin made an immense contribution by her X-ray crystallography data. Otherwise, it would have been difficult for Watson and Crick to really understand how these two helices are packaged, the role of hydrogen bond. So, the due credit to Rosalind Franklin was not given at the right time. People now realize her significance of work, but she's no more. So that's one of the science is sometimes is interesting, but you all being student, the reason I'm discussing this, that you being students and you have a long way to go, you should read literature or any story or any news very critically. There are a lot of things is going on, and particularly now with respect to social media and blogs and so many things. So when you read something, you should really critically think. That's the reason I dug this paper out, so to share with you. So that's the controversy part, and the reason I'm sharing the controversial part is that you develop critically, critical tone towards anything that you study. Science is definitely based upon facts, but there can be issues like this that I mentioned between Watson and Crick and Rosalind Franklin, and being a student of science, and you have a long way to go. You should strive for equity, equality, and credit to the person who does the work, right? So that's what, if things are based on facts, then they should be equal, right? Equal contribution, equal acknowledgement, equal awards and rewards. And people come to know 50 years back, but it's fine. But at the time, how would a person feel if the due credit is not given, right? So as a student of science, you have a long way. You should know these things and keep track of these things. So read here, this is very interesting. It says, we wish to suggest a structure for the salt of deoxyribose nucleic acid. See how he writes DNA, D period, N period, A period. Right, today's date, we just write DNA, but here it has been written. So that was the real original way that DNA was written, right? So now the first paragraph here starts with a comment on a previous paper by Pauling and Corey. They also published a paper on DNA, And their model suggested that DNA has 3 intertwined chains. Now be careful. They're not there, first of all, they're saying 3 intertwined chains. Secondly, they're not mentioning about helical structure, helicity in the DNA strands, right? So they say that, Watson and Crick says that the Pauling and Corey model consists of 3 intertwined chains with phosphates near fiber axis and bases outside, right? So Pauling and so you can imagine that the bases are inside, but the prediction of Pauling and Corey, they published their structure in 1953 itself, but they suggested three chains. They didn't say helical structure and the DNA bases pointing outside, which is just opposite. DNA bases are pointing inside. So Watson and Crick argue, they say that, now read here, we believe that the material which gives the X-ray diagrams is a salt, not free acid. So when you look at X-ray diagrams, what they are referring to? They're referring to 99%, I'm sure that they're referring to Rosalind Franklin's structures, X-ray, because she was an X-ray crystallographer in UK. And here they argue that without acidic hydrogen atom, it is not clear what forces would hold the structure together. Meaning that Pauling and Corey structure did not mention anything about hydrogen bonds. So if the hydrogen bonds are not there, how would the helical structure be, defined? So they're arguing. And then they say there was another 3 chain structure has also been suggested by Fraser, and this paper was in press. In this model, the phosphates are on outside and the bases are on inside, linked together by hydrogen bond. So this structure as described is ill-defined, and for no reason we shall not comment upon this. So, so I don't know, they are right, but what is the flaw here? That they again say that 3 chain structure, that DNA is not double helical, but 3 chain structure, but they predict that the bases are inside and they're joined by hydrogen bond. Now, after rebuttaling these 2, discoveries, Watson and Crick, they come back to their structure. And they say that we wish to put forward radically different structure for the salt of DNA, deoxyribose nucleic acid. This structure has 2 helical chains, just 2. Here it changes, right? Everybody before said 3 intertwined structure, but here he specifically mentions helical chain, right? And you should see here, these are the helical chains, which are very perpendicular to the axis, right? As if something is there central, like this line, and this is coiled, helically coiled. He says that they say that we have made the usual chemical assumption, namely that each chain consists of phosphodiester bond. So this is the first time they mention about phosphodiester bond, which was not mentioned in the earlier structure by Pauling and Corey as well as Fraser. And they say that this phosphodiester bond consists of phosphate diester groups joining beta-D-deoxy ribofuranose residues with 3,5 linkages, right? So if you just, I don't have the structure, chemical structure of DNA, I just have this model. If you go back and imagine the strand of DNA, phosphate is connected to fifth carbon atom and to the 3rd carbon atom between 2 sugar molecule, ribose, right? And the 2 chains are related by a dyad perpendicular to the fiber axis. So they're perpendicular on an axis wrapped around. Both chains follow right-handed helices. So they are very specific that helix, the direction of helix is right-handed, not. So when you're solving 3-dimensional structure, it is important to say whether it is right-handed or left-handed, right? So So these are the, now he comes here, it says very clearly that, see here, the thing that we discussed in the first chapter, or the first chapter of molecular biology, that there is a residue on each chain every 3.4 angstrom or 0.34 nanometer, which means that the distance between two bases is 0.34 nanometer or 3.4 angstrom. We have assumed that an angle of 36 degree between, so there's a major groove, minor groove, and you see here that this is a major groove and this is a minor groove, right? And there is a helix tilts upward by 36 degrees. So, so all these details have been mentioned in this paper very precisely. And then it says the novel feature of the structure is the manner in which the two chains are held together. They also gave the concept of purine and pyrimidines, which was absent in both the triple helix structures, triple chain structure. They say that purines base pairs with pyrimidine and pyrimidine base pairs with purine. And this helps to keep the diameter of the DNA constant throughout. And purine is single chain, purine is double chain, and pyrimidine is single chain. And there are triple hydrogen bonds and there are double hydrogen bonds. So very specific information. And all these specific information they got from Rosalind Franklin structures. And also defines for the first time that adenine and guanine are purines, and thymine and cytosines are pyrimidine. Right? So, so all these, and he talks about the previously published X-ray data, 5th and 6th. Yeah. So they talk about previously published X-ray data. So, so I wanted to share this original paper with you, and so that you get an idea what it takes to do something right. Yes, they were to the point with respect to the structure of DNA. It was 1953, and now it is 2022. It still holds. But the way science unfolded, especially when people knew that and came to know that the whole inspiration for this paper, which are the intrinsic details, hydrogen bonding. If you can't explain hydrogen bonding, how it is happening, you can't explain the structure of DNA. And to explain that, they had to take help of Rosalind Franklin structure, because the previous two structures were not able to explain, and they said that the two bases are connected by van der Waals forces. But this was very precise, to the point. Do you have any questions? So this was the paper on, so I wanted to start this chapter with history, right? And this being a molecular biotechnology class. So that was 1953. Now let's fast forward to 2022, right? Where are we in 2022, right? So in 2022, in April, right? It's very important when I'm teaching, I not only teach, but I try to keep myself for my own interest, know what's going on at this particular time. So now so this paper came out, let me tell you the exact date, sorry. This paper came out on April 1st, 2022. It's not a joke, it's serious, April 1st. So April 2022, 1st April, and fast forward from 1953, when the structure of DNA was. This is the complete, this paper shows the complete sequence of human genome. Now you can ask me, Professor, something was published in 2001, 2002, 2003. Yes, it was published, but it was not complete. The complete structure of a human genome, we only came to see now, right? So now instead of making slides, I am just showing you the original paper, and that will be, in this learning activity, this week's learning activity, I will not post any slides, just a couple of papers. So why I'm doing that? Because you can see the original work, and hopefully it will interest you to look at the authenticity of these work and read in your free time, and build your concept if you're interested in science, right? So this is 2022. Now let's read this. Let's read the abstract, right? Since its initial release in 2000, the human reference genome has covered only the euchromatic fraction of the genome. Euchromatic fractions are those fractions which are relaxed, right? Which could be active in making RNA, right? That RNA could be mRNA, tRNA, or rRNA, but mostly they might be protein coding genes. So heterochromatin was not finished, right? In 2000, the sequence that came out in 2000, the heterochromatin, the condensed region, like centromere or telomeres, they were not published, the sequence of DNA. So addressing the remaining 8% of the genome, the telomere to telomere, T to T consortium presents a complete 3.055 billion. See, if I teach, right, I can round it up, 3.1 billion or 3 billions, but precisely there are this much of base pairs in our 46 chromosomes, right? We are all diploid, 23 from mom, 23 from dad. So you can take this 3 billion, and I'm talking about ideal situation, and you can multiply between the bases. What's the difference? The difference between the two bases is 0.34 nanometers. So that would be the length of the DNA, right? And so also it says that human genome T to T, telomere to telomere, CHM13, that include gapless assemblies of all chromosome except chromosome Y. Corrects prior references, introduces 200 billion base pairs, right? So many information here, right? So the correct human reference genome was released by Genome Research Consortium, GRC, in 2013, and the most recent patch in 2019. So sometimes we, in a cell phone, we get updates, right? And those updates are to fill up the patchwork, right? Some patchwork between something has been, needs to be plugged in to make the running of cell phone efficient. So that's why you get once in a while update your iPhone, in the night, keep it plugged. So those are patchworks, right? So first of all, in 2001, only euchromatic DNA sequence was released. Then in 2013, some other batch came, and then the complete came in 2022, April 1. Now, this was the original human genome work. So what I would do, that I would just little bit fast forward. And I want to bring you to this table, right? And this table would compare the DNA sequence, the chromosomal sequence of DNA in 2013 and what it is released now. T2T, CHM13. So these are the differences. But before we go more detail, I would like to show you that what was published in 2001. And the reason I'm showing you is that you become familiar with a lot of vocabularies, right? So can you see these vocabularies? So you see, in the first or second chapter, we talked about various vectors that is used for cloning. So for human genome cloning, BSC was used. What is BSC? A bacterial artificial chromosome vector carrying a genomic DNA insert, typically between 100 to 200. So back clones were those clones which was used to create a library of human DNA, right? So so what is SNP, right? SNP is single nucleotide polymorphism, or a single nucleotide position in the genome for which two or more alternative alleles are present. So this is very important. You know, there's a lot of variation in human individuals. It could lead to disease, or it could be not part of a disease. But SNP, single nucleotide polymorphism, is very important to understand evolution. It's also important to understand the function of a protein, right? So I will like to go into little bit details of these. So I already set up a basic information about DNA as a starting point, and then we will cover what happened in excuse me, in 2001 very briefly, and then come back to the paper in 2022 that is published now. So that's the plan for the next class, which is Thursday. Okay. So I would like to stop here.